Today, I'll be discussing the recent advances in multiple sclerosis. I will start by introducing multiple sclerosis and its pathogenesis. Next, I will highlight the recent advances in genetic and environmental causes, as well as the new treatment methods used for MS patients. It is estimated that 2.3 million people suffer from MS globally, and at least 50% of these patients will pass away from MS-related symptoms. Individuals with MS constantly suffer from debilitating symptoms which greatly affect their quality of life. As a result, it is seen that suicide rates are increased in patients with MS compared to the general population, which highlights the importance of this topic. Multiple sclerosis is an autoimmune demyelinating disease affecting the central nervous system consisting of the brain and the spinal cord. Firstly, myelin is the protective sheath that surrounds the axon of neurons, providing quick and efficient electrical impulses along nerves. Myelin is produced by oligodendrocytes, which are a group of cells that support neurons. In multiple sclerosis, demyelination, the breakdown of myelin, is caused by immune cell infiltration across the blood-brain barrier. Initially, T cells make their way through the blood-brain barrier and inappropriately start attacking myelin as they are thought to be foreign. The attacking of these proteins release cytokines, which results in three consequences. Firstly, cytokines degrade the blood-brain barrier to be more permeable, allowing for more T cells into the CNS. Secondly, cytokines recruit B cells and macrophages into the CNS. B cells make antibodies that mark the myelin sheath, and the macrophages use these markers to engulf the myelin and the oligodendrocytes. Finally, cytokines are toxic to myelin and further destroy the myelin sheath. Initially, the breakdown of myelin sheaths can be repaired by the oligodendrocytes through remyelination, seen at the top. However, over time, after multiple autoimmune attacks, the oligodendrocytes are destroyed, resulting in neuronal death and the formation of lesions. These lesions can be detected on MRI scans and give a definitive diagnosis of MS. Furthermore, it is seen that these lesions lead to severe physical or cognitive disabilities as well as neurological defects. The cause of multiple sclerosis is still unknown. However, in recent years, neurologists have established certain genetic and environmental factors that contribute to its development. Recent studies comparing identical twins and fraternal twins have shown that the cause of multiple sclerosis has a genetic component. The study shows that if one identical twin has MS, the other with the same genetic makeup has a 30% chance of developing MS. In contrast, fraternal twins with different DNA only have 5% chance. Further research has shown that there are more than 50 genes that influence the development of MS. One gene in particular is of high interest, called the human leukocyte antigen, or HLA. HLA is used by the immune system to distinguish the body's own cells from foreign cells. This is achieved by the HLA protein on the surface of a foreign cell displaying a peptide in which the immune cells can scan and start to attack. In MS patients, HLA has malfunctioned as it highlights the myelin sheaths in the central nervous system as foreign, making the T cells form an attack. MS is also shown to have multiple environmental risk factors. Geographical location is of particular importance as vitamin D deficiency is seen to increase the chance of MS, as it is an important regulator of the immune system and can lead to autoimmunity. This is shown by the increased incident rate of MS away from the equator. These new understandings allow early detection of MS, which is crucial as it allows neurologists to administer the appropriate treatment methods before the debilitating symptoms occur. Finally, we will talk about the new advances in treatment. Current MS therapies are effective in reducing relapse rate, short-term disability, and inflammation in the CNS. Therefore, the focus of MS research has now turned to remyelination, which will restore the myelin sheath around the axon in the affected areas to counteract the autoimmune attacks. In the human body, oligodendrocytes are formed from oligodendrocyte precursor cells, or OPCs. However, in MS patients, it is seen that OPCs don't differentiate, meaning that they don't transition into oligodendrocytes. Therefore, an agent that can stimulate this transition will result in more remyelination, eventually reducing the impact of immune attacks and minimizing the debilitating symptoms, resulting in a better quality of life for MS individuals. The most suitable substance is clemastine, a first-generation antihistamine FDA-approved drug. It was discovered that clemastine had remyelination properties by Dr. Joanne Chang in his research of clinically used drugs and their impact on remyelination in an artificial setting. Recent studies by Dr. Ari Green and colleagues tested the effect of clemastine on 50 patients with relapsing multiple sclerosis with chronic demyelinating optic neuropathy. They found that those on clemastine had better conduction from the optic nerve to the brain, examined through a visual evoked potential test, compared to the placebo. It was concluded that clemastine promotes oligodendrocyte precursor differentiation and increased the chance of remyelination on damaged neurons. This new discovery gives great promise for the future treatments of MS patients.